What is going on, wonderful people? It's Mitochondria Perfectionalis, where medicine makes perfect sense. Welcome back to my physiology playlist. Today we'll have an introduction on physiology, which is the study of the normal bodily functions and the mechanisms of these functions. Today we'll learn about your cell and how your cell is organized. We'll talk about the nucleus, we'll talk about the cytoplasm, and we'll talk about the cytoplasmic organelles, such as the mitochondria, the Golgi apparatus, the rough endoplasmic reticulum, smooth endoplasmic reticulum, lysosome, peroxisome, and much more. We'll also talk about the types of transport. We have passive transport that does not require any energy, and we have active transport that does require energy. And always remember that about 2/3 of your body weight is made of water. Click the like button, click the subscribe button, and let's get started. In this physiology playlist, you will find more than 200 videos on every physiology topic that you can imagine. What does the word physiology even mean? Ology from logia or logos, the study of. Physis is the origin or nature or the mechanism. What's the definition of human physiology? It's the study of the functions of the human body and the mechanisms of these functions. Not just mechanisms, but regulations, positive feedbacks, negative feedbacks, stimulation, inhibition, and communication. Whether fast communication by means of neurons or slow communications by means of hormones. What is the basic building block of the human body? Of course, it's the human cell. How many cells do you have in your body? About 100 trillion. Anytime you find a number that is rounded and beautiful like this, it is not measured. It is estimated for the most part. No one, literally no one, counted all your cells. No one has the time or the capacity or the tenacity to do this. It's an estimate. Your body is a coordinated system. It has its own internal environment, or the milieu intérieur. About 60% or 2/3 roughly of your total body weight is made of water. Most of this water is in the intracellular compartment, meaning inside the cell. And the remaining third of that water is outside the cell in the extracellular compartment. So we have intracellular compartment inside the cells and extracellular compartment which is outside the cell. This extracellular compartment which is outside the cell is made of two subparts. First one is the interstitial space, which is interstitial between the cells or in between, and the plasma, which is inside the vessels. So the plasma is part of the extracellular fluid, and the interstitial space is also part of the extracellular fluid. But the fluid that's inside the cell, that's different. That's intracellular fluid. But what do we find in this fluid, specifically the extracellular fluid? Of course, you find water, you find electrolytes like sodium, like chloride, like bicarbonate, like calcium, like magnesium, etc. There is oxygen and carbon dioxide. There are nutrients. If you're eating proteins, we're talking amino acids. If you're eating lipids or fat, we're talking fatty acids. And if you're eating carbohydrates, we're talking glucose. We also have waste products, the good stuff and the bad stuff. Carbon dioxide is a waste. It is a product of metabolism that will be exhaled and eliminated by your lungs. This is volatile. And we have other garbage such as sulfuric acid, phosphoric acid, lactic acid, all kinds of acids, and these will be eliminated by means of kidneys. And this is non-volatile gunk. What's inside my cell? What is in the intracellular fluid or ICF? We have water and we have electrolytes. Pay attention. There is more sodium outside the cell compared to the inside. However, potassium is more abundant in the intracellular fluid compared to the extracellular fluid, creating ion concentration difference. This is the cell. This is the stuff that is inside the cell, and everything else is outside the cell. Sodium is more abundant outside the cell, but potassium is more abundant in the intracellular fluid inside the cell. Chloride is more abundant outside, which makes sense because chloride follows sodium to form sodium chloride. The negative is attracted to the positive. And this is Benjamin Franklin all over again. When he rubbed amber against cat fur or something like this and discovered the attraction between the positives and the negative. That was the story of static electricity. How about calcium? More abundant outside the cell. But magnesium is more abundant inside. Bicarbonate, more abundant outside. Phosphate, more abundant inside. Glucose glucose, more abundant outside. Proteins are everywhere, but they are more abundant inside because this is the cellular machinery. Don't forget that your pumps are proteins, your receptors are proteins, your channels are proteins, your carriers are proteins, etc, etc, etc. So how do I remember which electrolytes are more abundant intracellularly and which electrolytes are more abundant extracellularly? I just memorize the ones that are on the inside, and then the other ones are the other ones. What are the electrolytes that are more abundant in the intracellular fluid? All of them start with a P. You have potassium, you have proteins, you have phosphate, and we have magnesium, which is abundant in the supernova. That's another P. Everything else is more abundant outside the cell. Where do we get the word electrolyte from? From electrolysis. Oh, separation by means of electricity into positive charges and negative charges. Does anyone remember the cathode and the anode? Next, the story of homeostasis. If you want to see more videos like this in the future, please drop a heart emoji in the comments. Stasis means stable. Homeo means similar, constant, predicted, normal, balanced, etc. But hemostasis is not the same as homeostasis. Hemostasis is different. Hemo means blood, stasis means stable. In this case, it means the blood is not lost. The blood is clotting and not bleeding. If you want to learn more about hemostasis, I have a special playlist on this topic, and it's called Bleeding and Coagulation. Define homeostasis: maintenance of nearly constant circumstances in your body, in the internal environment. What's the internal environment in the body? Most students will say, oh, the internal environment is the intracellular fluid. That's a doofus mistake. The internal environment is the extracellular fluid because this includes your blood. Arteries, veins, capillaries, all of them are part of the extracellular fluid. And this is what homeostasis is all about. When we take a sample from your blood, do you think we're taking the sample from the intracellular fluid? No, from the extracellular fluid. It's easier to access the extracellular fluid than to access the intracellular fluid. Example of homeostasis. Look at your blood pH. Your blood pH is tightly regulated. Life is only possible within a very narrow range of change in pH. Your normal blood pH is 7.4 in the arterial blood. Less than 7, you're probably dead. More than 7.7, you're probably toast. So your blood pH better be tightly regulated. And this is what homeostasis is all about. Can we summarize physiology in one word? Sure, the word is homeostasis. Can we summarize pathology in one word? Sure. Disrupted homeostasis, lack of homeostasis, homeostasis-lessness. When your homeostasis is disrupted, congratulations, you are sick. There are people out there just like you and I who love studying human diseases. How does your body work? We have the good stuff, we have the bad stuff, and we need to communicate between the cells in your body. What's the good stuff? Well, I need food. Thank you, gastrointestinal tract. What else? I need oxygen. Thank you, respiratory tract. And I need to push that oxygen to every cell in the body. And to distribute that blood that has oxygen, I need the cardiovascular system. The bad stuff. Well, the liver is the hero of metabolism. Most of your biochemistry textbook takes place in the liver. And then the kidney will get rid of the non-volatile waste, whereas your lungs will get rid of the volatile waste. Communication within your body. We have fast communication and slow communication. Fast is neuronal, but slow is hormonal. Who brings us oxygen into the body? The lungs do. Now the oxygenated blood will flow from the lungs into the left side of the heart through these pulmonary veins. Now the oxygenated blood is in the left atrium, and then it will go to the left ventricle, and then the left ventricle pump it to the aorta, which will give oxygenated blood to every cell in your body, as you will see here. Each cell will take the oxygen and nutrients in, use them for metabolism, also known as internal respiration, and then dump the carbon dioxide and the waste onto the venous side. The venous side, which is now deoxygenated, will return back to the heart's right atrium. Here is the venous blood returning back to the right atrium. This is deoxygenated blood from right atrium to right ventricle. Right ventricle will pump this deoxygenated blood to the lungs. In the lungs, we'll have some beautiful gas exchange where the carbon dioxide is going to leave, hashtag exhalation, whereas the oxygen is gonna enter, hashtag inhalation. Your body's laboratory is the liver. It's the hero of metabolism of food and metabolism of medications. The liver is also an important storage unit for a whole bunch of stuff, including the famous vitamin B12. If you are a biochemistry connoisseur, you will remember that the urea cycle takes place in the liver, where ammonia, which is a waste of protein metabolism, is converted into urea. What do kidneys do? Kidneys filter the plasma. The good stuff will be returned back to you, hashtag reabsorption. The bad stuff will be dumped, hashtag excretion, into the urine. The kidneys help you regulate the acid-base balance, and the kidneys also have endocrine function, such as secreting renin. Why do we call it renin? IN because it's a protein, ren because it comes from renal, which is the kidney. Communication and regulation. Your nervous system, fast communication. Your endocrine system is slow communication. When a neuron is hugging another neuron, we call this synapse. The hormonal system is slow. We also call it the endocrine system. Endo means into, because all of these hormones will be dumped into the blood, into endocrine. Quiz time. The extracellular fluid, also known as ECF, has blank sodium ions when you compare it to the intracellular fluid. Please pause the video and let me know your answer in the comments. Next, the control systems of your body. The stories of the positive feedback and the more common negative feedback. In systems, we have feed-forward mechanisms and feedback mechanism. The feedback could be positive feedback or negative feedback. For example, if a rise in A causes a rise in B, and then the rise in B increases A, this is a positive feedback loop. Similarly, when a lack of A causes deficiency of B, and then the decrease in B causes decrease in A, this is another positive feedback loop. When the signs are going in the same direction, this is positive feedback. But when they are moving in opposite direction, this is negative feedback. And many students get confused here. When you tell a student that increase in something causes decrease in something else, they understand that this is negative feedback. But if you say to the same student, if a decrease in something increases something else, they tend to think, oh, this has to be positive feedback. Nuh-uh, not true. The signs are different. This is a negative feedback because the opposite to the initial stimulus is happening. When the opposite is taking place, it is negative feedback. If you wish to download these doozy colorful notes, go to Mitochondria.com. I help you learn, understand, and pass exams. If you want me to personally tutor you, reach out to me on my website. As for feed-forward versus feedback, it works like this. If the pathway goes X, then Y, then Z, X can feed forward by stimulating a step that will come later. But feedback is when an increase in Z causes decrease in a previous enzyme in a former step. Feed forward versus feed back. If you are a statistics buff, you would recall that this is similar to prospective cohort, but this is similar to retrospective cohort. Your body temperature needs to be tightly regulated. If your body temperature is too low, hypothermia, you're done. And conversely, if your body temperature is too high, hyperthermia, you're toast. And that's why your body is always talking about homeostasis, always buffering to prevent the sudden severe changes. For example, changes in pH. If your carbon dioxide increases in your body, carbon dioxide will grab some water, CO2 plus H2O will give me what? Will give me carbonic acid, which is H2CO3. Carbonic acid is an acid, of course. So your body needs to get rid of this excessive acid by breathing the carbon dioxide out of the body. Another important example of feedback mechanisms is the baroreceptor reflex. The word baro means pressure. So when your pressure goes up or when your pressure goes down, your body has to do something in response to this to bring the blood pressure back to normal. So if you started with hypertension, your body will try to reduce your blood pressure back to normal. And if you started with hypotension, your body, through several mechanisms, will try to increase your blood pressure and back to normal. If you want to learn more about the baroreceptor reflex, I have a special video for this topic, and you can find it in this physiology playlist. Calcium needs to be maintained at its optimum level or optimum concentration. Too little calcium, you develop tetany, which is carpal spasm, pedal spasm, laryngospasm, all kinds of spasms and convulsions and contractions. Conversely, too much calcium, your heart can stop during systole. Potassium needs to be tightly regulated. If potassium is too low, the heart will suffer from arrhythmias. Potassium is too high, the heart will suffer from different types of arrhythmias. Too much potassium can stop your heart during diastole. Too much calcium tends to stop your heart during systole, and too much potassium can stop your heart during diastole. Glucose. Glucose is too little, I can have seizure or coma. Glucose is too high, I can also develop seizure and or coma. That's why homeostasis is paramount. If for whatever reason I have too much carbon dioxide in the body, my body will respond by breathing rapidly or hyperventilating to wash away that carbon dioxide, bringing it back to normal. Conversely, if I started with hypocapnia instead of hypercapnia, my body will breathe slowly to keep the carbon dioxide in. Carbon dioxide keeps building up and building up and building up inside my body to normalize and to correct the original insult. Positive feedback versus negative feedback. Which one is more common in your body? Negative feedback is. What's the problem with positive feedback? Positive feedback is explosive. Let me tell you one example of why positive feedback is mostly evil. Suppose that I lost 2 liters of blood. Now my heart is pumping less blood because my circulatory system is having less blood. This means that less blood is going to the aorta, less blood is going to my coronary arteries which supply the heart. So now the heart will be supplied with less blood, so the heart will pump less, which will supply it with even less blood, which will make the heart pump less. This is a positive feedback loop. It's a vicious cycle that will end up with my demise. But positive feedback is not always evil. It can be actually beneficial. For example, if I lose some blood, tiny drops, I will activate my clotting cascade or coagulation cascade. This will act on other enzymes in the vessel to constrict the vessel. Constriction triggers more coagulation or makes it easier, which makes more constriction, more coagulation until the bleeding stops. So this is a good example. Another good example is uterine contractions to push the baby out. Mom will give the baby a push, and then this push stretches the uterus smooth muscles even more. More stretch triggers more contraction. More contraction pushes the baby downstairs, stretching the uterus yet again, which triggers more contraction. Contraction pushes down, pushing down triggers stress, stress triggers contraction. This is a beautiful positive feedback loop, which ends up in delivery of the newborn. If you want another example of positive feedback loop, it is the first half of the menstrual cycle, known as the proliferative phase. And I've talked about the menstrual cycle in detail in my reproductive physiology videos, which you can find in this physiology playlist. Now let's talk about your cell, which is an animal cell, not a plant cell. Your cell does not have a cell wall, but it does have a cell membrane, also known as plasma membrane, also known as lipid-bilayer membrane. You have trillions and trillions of cells. You, as a human being, are a eukaryote. Eu means true, karyote means nucleus. You have a true nucleus, bound by and surrounded by a true nuclear membrane. That's why you are eukaryote. You are not a primitive karyote. Bacteria, for example, are prokaryotes. They do not have a membrane around the nucleus. That's why we say it's a primitive nucleus. Prokaryotes do not have mitochondria, but you do have mitochondria. And because your liver is super duper active, because the biochemistry lab is your liver, each liver cell has thousands of mitochondria. Prokaryotes, like bacteria, do not have membrane-bound organelles, but you do. Bacterial DNA is naked and circular or linear, but your DNA is protein-bound, we call these histones, and your DNA is linear. It's a double-stranded, anti-parallel, double helix. Watson and Crick have entered the chat. Some prokaryotes have only one cell, but your body has many cells, multicellular. To learn more about mitosis versus meiosis, please refer to my biology playlist. Pro means before or primitive. Karyote is a nucleus or a nut. Eukaryote, true nucleus or true nut. So instead of saying to your body, hey, you drive me nuts, it is more accurate to say, what's up, you're driving me karyons. What are the 4 fundamental tenets of the cell theory? Number 1, all living things are composed of cells. Number 2, the cell is the basic functional unit of life. Number 3, cells arise only from pre-existing cells. And number 4, cells carry genetic information in the form of DNA. The building unit, the building block of your body is the cell. A bunch of cells together, a tissue. A bunch of tissues together will form an organ, and a collection of organs will form a system, and the systems will carry body functions. So your cell has a nucleus. This nucleus has a nuclear membrane around it, and your organelles also have a membrane around them. This is your cell, this is the nucleus, and this is the cytoplasm. Nucleus plus cytoplasm equals protoplasm. Your cellular organelles are in the cytoplasm, and your cell membrane is a lipid-bilayer. Is there a difference between the cytosol and the cytoplasm? Yes, there is. And I have a special video on this topic. You can find it in my biology playlist. Form follows function, not the other way around. For example, sperms need to move a lot. They need to swim. They need lots of energy. They need lots of mitochondria. In fact, the middle piece of the sperm contains mitochondria. The islets of Langerhans in the pancreas need to secrete. Secrete what? Insulin, glucagon, and somatostatin. And in order to secrete, you need the Golgi apparatus and rough endoplasmic reticulum. They work hand in hand. I've talked about Golgi in a separate video and about the endoplasmic reticulum in its own video. You can find them in this physiology playlist. Red blood cells need all the space in the cell just to carry oxygen and or carbon dioxide. So therefore, the red blood cell does not have organelles. The red blood cell does not even have mitochondria. Therefore, if you are a biochemistry connoisseur, you'll recall that the tricarboxylic acid cycle, known as the Krebs cycle, and the electron transport chain take place in the mitochondrion. But since the red blood cells do not have mitochondria, therefore the red blood cells do not have Krebs cycle, and they do not have electron transport chain, therefore they do not undergo oxidative phosphorylation. Their only hope for energy comes from glycolysis. To learn more about glycolysis, Krebs cycle, and the electron transport chain, please refer to my biochemistry playlist. Next, cytoplasmic organelles. What are the functions of your cellular organelles? The mitochondrion is the power plant. It provides energy for your cell in the form of ATP or adenosine triphosphate. The lysosome is the destroyer. It destroys foreign invaders, and it destroys your debris that needs to go. The endosome is the delivery guy. It makes packages and packages the stuff and delivers these boxes. The endoplasmic reticulum comes in 2 shapes. Those with ribosomes, rough endoplasmic reticulum. It is rough because it's studded with ribosomes. And the ones with no ribosomes, so they are smooth because there is no ribosome in them. So rough endoplasmic reticulum and smooth endoplasmic reticulum. The rough endoplasmic reticulum is the translator. What does that mean? It translates proteins for secretion. Translation means protein synthesis. But the smooth is smooth, like your belly. Like a donut, fat synthesis. Golgi is a sorter. It's an organizer. It modifies the products of the cell, sorts them, and delivers them. The peroxisome is a gym trainer. It is a fat destroyer. Let's start with the mitochondrion. It has an outer membrane, it has an inner membrane, it has an intermembrane space, and it has a mitochondrial matrix. The mitochondrion is grain-like, and that's where the name comes from. The singular is mitochondrion, plural mitochondria. The mitochondrion is the power plant of the cell. It makes ATP, which is the energy currency. It's not just an energy producer, but it's also a murderer. What kind of murder? Are we talking homicide or suicide? We're talking suicide. Hashtag apoptosis. If you are a student of pathology, you will recall that we have 2 types of cell death. apoptosis, cell suicide, and necrosis, which is cell homicide. Your nucleus has its own DNA, and your mitochondrion has its own DNA. The mitochondrial DNA is circular and with no histones, but your nuclear DNA is linear and it does have histones. The functions of the mitochondria are numerous. ATP production, thank you Krebs cycle, thank you electron transport chain and oxidative phosphorylation, cell signaling, cell differentiation, control of the cell cycle and cell growth, heat production. Does anyone remember the brown fat? I've talked about this in my metabolism videos, which you can find in this physiology playlist. Heme synthesis, and it's a killer. Your mature red blood cells do not have a nucleus. They do not have mitochondria either. Conversely, each liver cell has thousands of mitochondria. Your mitochondria produce most, but not all, of the ATP for the cell. Remember, this is mommy, this is daddy. Your daddy left his mitochondria outside. Therefore, your mitochondria in your cell are maternal in origin. They came from your mother, not from your father, because your father left his mitochondrion outside. He left his tail outside as well. The moral of the story is the mitochondria is not just for energy. Mitochondrion is also a killer. Give me liberty or give me death. Dear mitochondria, give me energy, ATP, or give me death, apoptosis. The next organelle that we'll talk about today is the lysosome. Some from soma, which means body, hence somatic. Also the soma in the neuron. And lyso from lysis, which means breakdown, which means destruction. The lysosome is membrane-bound organelle. No kidding. It destroys by means of hydrolytic enzymes that work in an acidic medium. The lysosome can help you kill foreign invaders as well as endogenous stuff that needs to die. For example, when this cell is very old and malfunctioning and it's time for it to go by apoptosis, guess who's gonna kill your own cell? Your lysosome. It's called autolysis. The lysosome works hand in hand with an endosome. That's why we can say endolysosome. It also works hand in hand with a phagosome. Hashtag phagolysosome. These hydrolytic enzymes are catabolic. They break down. They do not build up. When you break down proteins, they become amino acids. When you break down triglycerides, they become free fatty acids and glycerol. When you break down carbohydrates, they become glucose. For example, glycogen becomes glucose. What does hydrolysis mean? It means lysis in the presence of hydro, breakdown in the presence of water. What does the lysosome digest? Bacteria and other stuff, ingested food particles, and cellular structures that were damaged. Look at this, look at this, look at this endosome, and this is your lysosome, and they work together. Why do we call it endosome? Because it's a body of endocytosis. We'll talk about endocytosis later in this video. Next, it's time to talk about the endoplasmic reticulum, the Golgi, the peroxisome. The endoplasmic reticulum, why do I need it? It's the translator for protein synthesis and secretion. The endoplasmic reticulum is continuous with the membrane of the nucleus. We have two types, rough endoplasmic reticulum and smooth. The rough is granular because it has ribosomes. The smooth is non-granular because it does not have rhizosome. The rough, since it has ribosomes, is important for translation, which is protein synthesis. But the smooth deals with fat synthesis, as well as detoxification. This is rough endoplasmic reticulum because it's studded with ribosomes. But this is the smooth endoplasmic reticulum. Protein, fat. Rough endoplasmic reticulum is needed for processing proteins, transporting proteins. It has ribosomes. Remember that ribosomes are made of what? RNA and proteins. The smooth endoplasmic reticulum is for fat synthesis, including the phospholipids of the cell membrane and steroid hormones. Can you give me examples of 3 different steroidal hormones? Please comment below. Next, Golgi, which works hand in hand with the rough endoplasmic reticulum. What is Golgi? It's a bunch of membrane-bound sacs on top of one another. Stuff comes from the endoplasmic reticulum to vesicles to Golgi, and then Golgi pushes them out. Golgi is a sorter and a modifier. This is your nucleus, and this is your nuclear membrane, which is continuous with the membrane of the rough endoplasmic reticulum. Make some vesicles, push them forwards. Make some vesicles, push them forwards, and then secrete them. These are called secretory vesicles. You push them out by a process called exocytosis. Cyto means cell. Osis means condition. Exo means exit, extrusion. Push it out. Form follows function. If your cell needs to secrete lots of stuff, such as the Langerhans cell in the pancreas, therefore these cells will contain lots and lots of endoplasmic reticuli and Golgi apparati or apparatuses. What's the plural of apparatus? Let me know in the comments if you know. Next, the peroxisome. Do you remember hydrogen peroxide? That's the same origin. Again, some from soma means body. Peroxisome, oxy, they contain oxidase enzymes. They contain hydrogen peroxide. This is the gym trainer because it breaks down fat. Breaks down fat how? By beta oxidation. To learn more about beta oxidation and the peroxisomal diseases, please refer to my biochemistry playlist. We are done with your cellular organelles. Please take a moment to pause and review. Next, let's talk about your cell membrane. Here is your cell membrane. Sometimes we have proteins that take the entire thickness of your cell membrane. We call these integral proteins. They could form pores or channels. Some channels are gated with a gate, sometimes are not gated. Remember that oil and water do not mix. Remember that outside of your cell there is water. Inside of your cell there is also water. And that's why we needed a lipid bilayer in the membrane. Lipid bilayer. Why bilayer? Because these heads are hydrophilic, they love water, and they are facing water. That's nice. These heads are also hydrophilic, loving of water, and they are facing water. Amazing. But in the middle, the tails are hydrophobic. They hate water because they are water insoluble. This is a lipid bilayer membrane. Now you understand why this membrane had to be lipid bilayer. Not one layer, but it had to be two. So that you're facing the water outside and you're facing the water inside the cell. What if there is tiny stuff that needs to go out or go in? We can have holes in the membrane. We call them pores. Even your nuclear membrane had pores. Next, the cytoskeleton. So not only your body has a skeleton, but each cell of your body has its own cytoskeleton, the skeleton of the cell. Because why not? The most abundant thing in your body is water. The most abundant thing in your cell is also water. The second most abundant is protein. We have many types of proteins, structural and functional. The proteins that make the cytoskeleton are structural proteins because they help lay down the foundation of the cell, the infrastructure of the cell. Structure, not function. Why do we need a skeleton in the cell? To determine and establish a shape for that cell, to help in cell division and in movement, and in cell division into two. Also, it provides a tram track for the organelles to move on. Examples of cytoskeleton: actin filaments in the ectoplasm, actin and myosin in your muscles, tubulin in the microtubules. Cytoskeleton functions: provide structure involved in muscle contraction, tram tracks for movement and migration, cytokinesis, cell signaling pathway discussed in my endocrinology playlist, separate my chromosomes during cell division, #mitosis and meiosis 2. Please refer to my biology playlist and provide a template for cell membrane construction. There are three types of cytoskeleton: microfilaments, microtubules, intermediate filaments. Microfilaments, they are made of actin. Microtubules, like the cilia and flagella, they are made of tubulin. Tubulin, tubules. And intermediate filaments. The structure of the cilia or flagella look like this. You have two central single microtubules in the middle, and then you have 9 peripheral doublets with the famous dynein arm. Can you tell me what is the name of the disease where there is a defect in the dynein arm? If you know the answer to this question, please comment below. 9 doublets and 2 additional central microtubules is cilia or flagella. But 9 triplets of microtubules is the structure of the centrioles. And that's why you need cytoskeleton to help you during cell division. Microfilaments, microtubules, and intermediate filaments is what the cytoskeleton is all about. Intermediate filaments include many. Vimentin is found in mesenchymal cells, which are mesodermal. Keratin in epithelial cells, neurofilaments in neural cells or neurons, neuroglial in neuroglia, lamine in the nuclear envelope, desmin in the muscle. Let's talk more about the cell membrane. It is a lipid bilayer, as you know. You have a cell membrane, but you do not have a cell wall. Your cell membrane is also known as plasma membrane or lipid bilayer membrane. Your cell membrane is elastic, it's dynamic, it is semi-permeable. Some stuff is allowed to enter, others are not. Your cell membrane is made of proteins, 55%, lipids, composing 42% of the membrane, and carbohydrates, only 3%. Types of lipids in your cell membrane include phospholipids, cholesterol, and sphingolipids, especially in nerves. To learn more about sphingolipidosis and other lysosomal storage diseases, please refer to my biochemistry playlist. Why do I need a cell membrane around the cell? It's a barrier. It's a medium for passage. It is semi-permeable. We call this selective permeability. If you are water soluble, you cannot pass through the lipid membrane. But if you are lipid soluble, you can pass easily. But hey, I am water soluble. Is there hope for me? Yeah, we can create some channels or holes in the membrane to allow you in and out. The cell membrane lipids have hydrophilic heads and hydrophobic tails, as we have discussed before. The heads are on the periphery, whereas the tails are central. The heads are negatively charged. Why? Because this is phospholipids. This is phosphate, and remember that phosphate is negatively charged. If you are a chemistry geek, you would recall that if a word ends in ate, odds are it is negatively charged, such as bicarbonate, sulfate, phosphate, and others. As for the tail, it is hydrophobic. It's made of free fatty acids. The head has a negative charge, but the tail does not have any charges. That's it for the lipid in the lipid membrane. How about the proteins in my cell membrane? You have integral proteins that take the entire freaking thickness, and you have peripheral proteins. They are peripheral. Some of them are on the outside, such as receptors. Some of them are on the inside, such as enzymes. Since metabolism occurs inside the cell, and metabolism needs enzymes, therefore enzymes are on the inside. Since hormones float in the bloodstream, hormones touch the cell from the outside, so hormones need receptors. We're going to put receptors on the outside. Enzymes on the inside, receptors on the outside. All of these are proteins. Your enzymes are proteins. Your pumps are proteins. Your receptors are proteins. Your channels are proteins. Your carriers are proteins. Every active thing in your body is protein. Your plasma membrane is made of proteins, lipids, and carbohydrates. Carbohydrate is for ID, for recognition sites, and for adhesion. Around everything there is a glycocalyx. It's a sugary coat made of carbs because sugar is sweet. The active component in your body is protein. The more active the cell, the greater the protein content. Next, let's dig deeper into the nucleus, which is the command center of the cell. I can see a lumen at the center of the cell. He was talking about the nucleus. That's the story of Antony van Leeuwenhoek. Again, what does the word nucleus mean? It means nut. The nucleus is the control center of the cell. The nucleus contains your genetic material, which is DNA. Viruses have DNA or RNA, but humans have DNA as the genetic material. From this DNA, you can make RNA if you want, a process known as transcription. You can learn more about transcription and translation and replication and modification and spliceosomes and others in my biochemistry playlist. In humans, your nucleus is surrounded by a nuclear membrane that has pores in it. Inside your nucleus, of course, you have your DNA, the double helix, which is wrapped around histones, tightly packed and wrapped into chromosomes. Each somatic cell in your body has how many chromosomes? The answer is 46, except the gametes, ova and sperms. The ovum has 23 chromosomes, and of course the sperm has 23 chromosomes, so that a piece from mommy and a piece from daddy, they combine together to make you with a 46 chromosome. Autosomal versus sex cells. What does replication mean? Replication means to take that DNA and double it. Make another copy. This is replication. What is transcription? To convert DNA into RNA, usually mRNA. Then mRNA is going to exit the nucleus through the nuclear pore and go to the cytoplasm. In the cytoplasm, translation will take place. And translation is when you convert the mRNA into proteins. Transcription takes place in the nucleus, but translation happens in the cytoplasm. And that's why the membrane of the nucleus is continuous with the membrane of the rough endoplasmic reticulum, because we're talking about proteins here. See, medicine makes so much sense once you understand what the French toast you're talking about. Inside your nucleus, there is a nucleolus. Your nucleus has a membrane. It's called nuclear membrane, but your nucleolus does not. Why do we need a nucleolus? To make your rRNA, the ribosomal RNA. Most cells of your body have only one nucleus each. Red blood cells do not have a nucleus to allow more space for oxygen carrying capacity. The osteoclasts of your bone have multiple nuclei. White blood cells, especially neutrophils, you can have segmented nucleus. It's one nucleus, but it's made of many lobes or many segments. If your neutrophil has too many segments, this is a disease known as megaloblastic anemia, which could be caused by vitamin B12 deficiency or folate deficiency. Next, we'll talk about the very important topic of cell membrane transport. There are two types of transport. There is passive transport and active transport. The passive transport is passive because it does not require any ATP, and it happens along the electrochemical gradient. That's why there is no need for energy. But the active is called active because it does require energy. Passive has many types, including simple diffusion, osmosis, and facilitated diffusion. By the way, osmosis is a subtype of simple diffusion. Active transport has two types, carrier transport and vesicular active transport. Let's start by talking about the vesicular active transport, which has two types, endocytosis and exocytosis. Endo means into, exo means exit. Let's talk about it. How does the cell get its nutrients? Well, it depends. If we're talking water, water can pass quickly through channels for water called aquaporin. I love the name. Why does it end in IN? Because these channels are proteins. Protein end in IN. Why aqua? Because it's for water. And pore because it's a hole. It's a pore in the wall. It's a pore in the cell membrane. If you are a water-soluble particle, you need channels or pores. If you are lipid or lipid-soluble, you can pass through the lipid-bilayer membrane. But what if you are a macromolecule? What if you are big? Then you need vesicular transport, such as endocytosis or exocytosis. But since the cell is trying to eat, we're only concerned with endocytosis right now, which has two types, pinocytosis and phagocytosis. Pino means to drink, for small particles. Phago means to eat, for bigger particles. These first three are examples of passive transport that does not need any energy. But macromolecular vesicular transport, whether it is endo or exo, phago or pino, all of these are active transports, which means they require energy. Vesicular transport is a type of active transport, which means it requires ATP, and it has two types, endocytosis and exocytosis. Endo means going into the cell. Exo means going out of the cell, to exit. Endocytosis is subdivided into pinocytosis, smaller particles, phagocytosis, relatively bigger particles, but make no mistake about it, both of these are for macromolecules. So both of them are big, but the phagocytosis ones are bigger. Next, we'll talk about passive transport, which does not require ATP, which occurs along the electrochemical gradient, meaning from higher concentration to lower concentration. And the negative will be attracted to the positive, or the positive to the negative. This passive transport is also known as diffusion. It has simple diffusion, osmosis, and facilitated diffusion types. Osmosis is a special situation of simple diffusion. Osmosis, it's just simple diffusion for water. Simple diffusion does not need any energy, occurs along the electrochemical gradient from higher concentration to lower concentration of that solute. For example, sodium will go from higher concentration of sodium to lower concentration of sodium. Not only does it require no energy, but it also requires no carrier either. Simple diffusion can happen through the lipid membrane if you are a lipid-soluble molecule, through channels, whether gated or non-gated, and if you are a gated channel, some gates open with electricity, we call them voltage-gated, some gates open with chemistry, we call them ligand-gated. The ligand is something that binds to the receptor like a key in a lock, like a truck in a dock. Molecules that are small and water-soluble need channels to pass through the membrane. Water is unique because water can pass through the membrane slowly, or it can pass through aquaporin channels quickly. Let's talk about diffusion, which does not require ATP. Diffusion is passive. It does not require ATP. It occurs from higher concentration to lower concentration. But active transport is the opposite. It does require energy, and it happens against the electrochemical gradient, meaning from low concentration to high concentration, and that's why you need energy. What is osmosis? Osmosis is a special type of simple diffusion. It's simple diffusion just for water. What is simple diffusion of sodium? It's from high concentration of sodium to low concentration of sodium. Similarly, the simple diffusion of water is from high concentration of water to low concentration of water across the membrane. So here is this semi-permeable membrane, and we have sodium particles on the left, okay, and we have sodium particles on the right as well. Notice that compartment A has more sodium particles than compartment B, so therefore there is more water in compartment B and less water in compartment A. So water will pass from high concentration of water to low concentration of water. This is osmosis. You can say this in a different way. You can say that water is flowing from low concentration of sodium to high concentration of sodium. So you got to be very careful whether you're talking about water or sodium. Compartment A has more solute. Compartment B has less solute. Put differently, compartment A has less water and compartment B has more water. Therefore, if this is a semi-permeable membrane, water will be transported from higher concentration of water to low concentration of water, or from low concentration of sodium to higher concentration of sodium. Put differently, the higher concentration of sodium attracts water towards it because sodium is osmotically active. Next is facilitated diffusion. It is still passive, so there is no need for ATP. It is still passive, so it occurs along the electrochemical gradient from high concentration to low concentration. But unlike simple diffusion, it does require a carrier. And because it requires a carrier, the carrier is going to give it three characteristics: structural specificity, competition, and saturation. What does structural specificity means? It means that you need one key for one lock. The ligand needs to be specific for that receptor. Not any ligand can bind to any receptor. Next, competition. You can have two molecules competing for the same receptor if they fit to that receptor. Next, because you need a protein carrier, in this case it's the receptor, eventually you will run out of receptor and you will reach saturation. So when you had enough receptors, you kept increasing with increasing concentration. The more concentration, the more the diffusion, until you have saturated all of your protein carriers, and then you're gonna plateau. You're gonna level off. You're gonna reach your maximum, we call it Vmax, the maximum rate or the maximum speed of diffusion. Next, let's talk about active carrier transport. Mnemonic for the pros. Diffusion happens down gradient from high concentration to low concentration, but active transport occurs against gradient. But facilitated diffusion, well, I am simple diffusion, but I need a freaking carrier. And that's why I'll be saturated and reach my roof, the flippin' Vmax. Active transport, does it require energy? Yes. Does active carrier transport require a carrier? Yes. In this case, it's like ATPase. Active transport requires energy in the form of ATP. We have two types of this: carrier-mediated and vesicular. Carrier could be primary active transport or secondary active transport. The vesicular could be endocytosis or exocytosis. Endo is into and exo is exit. Endocytosis is subdivided into phagocytosis for very, very, very large molecules and pinocytosis for smaller molecules. Active carrier-mediated transport. We have two types, primary active and secondary active. Both of them require energy in the form of ATP, and therefore we need an ATPase activity. What's the difference between primary active or secondary active? Primary active exerts energy itself, but secondary active is dependent on the primary, just like this guy is dependent on that guy. The greatest example is the train going uphill. Primary active transport is here, where the engine is located. This is where the fuel is actually burned. Therefore, primary active transport needs ATP, and secondary active transport needs ATP as well. But this energy was not burned and released here, but here at the primary active site. And then the secondary is gonna hitchhike and piggyback and utilize the energy released by the primary active transport. Classic example is this story: the primary active transport, which is sodium potassium ATPase, and the secondary active transport, which is sodium calcium exchanger. Primary active transport, it's sodium potassium ATPase. You're going to push sodium outside the cell and push potassium into the cell. You are pushing sodium out, pushing sodium out, pushing sodium out until sodium accumulates outside. All of these are positive charges and they repel one another. Sodium becomes sick and tired of being sick and tired, so they repel one another and boom, sodium starts to enter back into the cell. Sodium is positive, and to maintain electroneutrality, you gotta push something positive out. If something positive is going in, something positive has to go out. In this case, it's sodium versus calcium, and we call this a sodium-calcium exchanger or a sodium-calcium antiport. Port from la porte or la porta, which means door. The door that has different or opposite directions. Sodium potassium ATPase is a classic example of primary active transport, whereas sodium calcium exchanger is a classic example of secondary active transport, which means if the primary ATPase is blocked, such as by taking a medication known as digoxin, then the secondary active transport will also be blocked because the secondary is dependent on the primary. Digoxin blocks the primary active, then the secondary active is also stopped. Therefore, calcium is not going to leave, and calcium will accumulate inside the heart cell. Remember that calcium is the hero of contraction, so your heart muscle will contract more. And that's why digoxin can help some patients with heart failure. Because in heart failure, my heart does not contract as much. Therefore, I'm going to give digoxin, and digoxin is going to make the heart contract harder. To learn more about digoxin, please refer to my pharmacology playlist. To learn about congestive heart failure, please refer to my cardiology playlist. Please take a moment to pause and review. To learn more about this equation for the osmotic pressure, please refer to my general chemistry playlist. Please recall that osmosis is a colligative property. It only cares about the number of particles, not the mass, not the size, just the number. Here is another question for you. Here's compartment A and compartment B before equilibrium. What do you think is going to happen to the water level at equilibrium? Is it going to be like this, like this, like this, D, E, or F? Pause the video and let me know your answer in the comments. And here is one more. Calculate the osmolarity of the solution in each case, then write the answer in the corresponding space. In today's video, we learned about homeostasis, feed forward and feedback, whether it's positive feedback or negative feedback. We talked about your cell. We talked about the cell membrane. We talked about the cytoplasm and the organelles of the cytoplasm. We talked about the nucleus and the nucleolus. Then we talked about cellular transport. There is passive, there is active. Passive transport includes simple diffusion, osmosis, and facilitated diffusion. Active transport includes carrier active transport, primary and secondary, as well as vesicular transport, endocytosis and exocytosis. Now, enjoy the rest of this physiology playlist. Are you struggling to understand kidney physiology? The glomerular filtration rate, the drama that takes place in the proximal tubule, loop of Henle, distal tubule, and collecting ducts? Download my kidney physiology course at medicosisperfectionalis.com, which comes with videos, notes, and cases. If you value what I do, help me make more videos by supporting the channel. Go to buymeacoffee.com/medicosis. There are more than 600 premium videos available on this channel when you click the join button and choose the highest tier. Please subscribe, hit the bell, smash like, support my channel on Patreon, PayPal, or Venmo. Go to my website to download my courses, notes, and cases, or if you would like me to personally tutor you. Be safe, stay happy, study hard. This is Medicosis Perfectionalis, where medicine, chemistry, math, and physics make perfect sense.